Well, thank you very much for inviting me to do this talk. I thought I would take you on a little bit of a journey through time and space, although not in a TARDIS, I hasten to add. And in doing so, I'd really like to use this as a bit of an opportunity to pay tribute to the many people that I've worked with over the past, well, it's actually longer than 4 decades, the students, the colleagues, and many of whom have been instrumental in KRAG as well. I did think when I started, it's about half of the work that I've been involved with has been sort of locally based and about half overseas. And I did think about including some of the overseas work, but I'm afraid that I would start rambling and you probably wouldn't have time for tea if I started on that. So I'm very much going to confine this to activities that I've been involved with Kent and other parts of the UK. So taking you back to the very beginning, I'm always interested in sort of how people get interested in natural history and in herpetology. And this young man here may look vaguely familiar. This was a 10-year-old Richard Griffiths on a family holiday to the Isle of Wight Zoo. And this really was the first close encounter with a snake that I'd had. And this was a boa constrictor from the reptile house. And this, you know, without sounding too cheesy, it probably changed my life. After this event, I was incessantly badgering my long-suffering parents that I wanted a pet snake. They steadfastly refused that because my mother said if I brought a snake into the house, then she would leave immediately. But they were a little bit more sympathetic. And a little bit later on, I started keeping newts and frogs and things like that. Now, you're no doubt wondering who this gentleman is on my left, who's clad only in a fairly sort of scruffy pair of trunks. Well, this was somebody who went by the stage name of Nick Nyoka, and he was a fairly well-known zoo circus man of his generation. And his hallmark was having big animals. And at the Isle of Wight Zoo in those days, he had what he claimed was the biggest lion in the world, called Simba, the biggest tiger in captivity, and also the biggest snake in captivity. And hopefully you can see this rather grainy photograph of this reticulated python that was apparently 27 feet long and went by the name of Cassius. And Gerald Durrell was very much a strong influence in my early years, and Gerald Durrell wrote this comment in an introduction to a book in 1994 that every herpetologist that he'd ever met had begun life as a mini zookeeper, keeping things. And I make no apologies for the fact that that's exactly how I started, keeping frogs and newts and other small creatures like that in my bedroom. But as far as my parents were concerned, definitely drawing the line at snakes. Being brought up in London, I didn't live that far from London Zoo, and somewhere that was very instrumental in influencing my development was the reptile house. I visited there very, very frequently. And of course, today, I think the reptile house is on the Harry Potter tour of London, because you can go and see where I think, was it Dudley Dursley fell into the snake enclosure after Harry made the glass disappear after talking in parcel tongue to the snake there. But for me, the interesting thing about London Zoo was something that isn't there anymore. In front of the reptile house in the 1970s was an outdoor reptillery. This was bulldozed when I think they built the new primate enclosure. But this outdoor reptillery had native reptiles on it. And I spent many an hour trying to spot the reptiles there. There was a low wall with a little moat around the inside. And I remember seeing an adder swimming in the moat. And I thought that was the first time I'd ever seen an adder. And also I thought, well, that's interesting because none of the books I've read say that adders swim very well, but clearly they do. So this was my sort of upbringing in London in my early days. And then the 1970s. Now the 1970s are thought of as a decade when style and fashion sense had a bit of a wobble. And I think this is epitomised by my school photo taken here sometime in the middle of the 1970s. I'm down here in the front as I always was in school photos, not because I was an egomaniac, but simply because I was the shortest boy in the year, and the teachers always pushed me to the front because they thought if I stood anywhere else, I'd be invisible in the photo. But by this stage, my sort of herpetological apprenticeship had developed a little bit. I was never very sporty at school at all, but I did get into the school cross-country team. And I think that was purely on account of the fact that I'd learned to run quite fast on account of being chased by park keepers and golfers who discovered me netting their ponds that they're responsible for. But the other thing that I do remember at school was I was sitting in the library one Friday afternoon trying to finish a particularly tedious essay for our English master. And I glanced across to the magazine rack that was in the corner, and there was a copy of that week's New Scientist magazine there. And it had a fantastic photograph of a smooth newt on the cover with the headline, "Playboy of the Watery World." So looking for a distraction from this tedious English essay, I picked up that magazine and read it, and I was absolutely enthralled. It was an article written by a young herpetologist by the name of Tim Halliday, you may have come across, and at the time I didn't realise it, but a few years later, I get to know Tim Halliday very, very well. And 2 years ago, when Tim sadly died, I had the honour of writing his obituary. But this was when I was at school, and I read this article and was absolutely entranced by it. And I put it down, and then I started doing some strange doodles on the cover of this magazine. And then, of course, I got caught by my English master, who came round and said, don't seem to be making much progress with your essay English. What's this you're reading? And I said, well, it's a very interesting on newt behaviour, sir. Now, in those days, schoolmasters' main tools of the trade for keeping you in line was sarcasm and humiliation. And he noticed the doodle that I put on the cover of this magazine. I'd converted New Scientist to Newt Scientist. And of course, he picked this up and said, well, Griffiths here thinks he's going to be a newt scientist when he gets older. And everyone sort of sniggered. And he pointed out to me that I'd defaced school property and that I needed to replace this copy of New Scientist in the school library, which I dutifully went out and did. But I kept the old copy, which is why it's appearing in this presentation here. But my interests in newts and amphibians were developing right through school, and then took another step forward when I went to university. And I went to university at a place called Westfield College in London, and I lived here for 2 years, and much of my biological education occurred here. But what was particularly of interest was that there are a number of ponds in the grounds of Westfield College. And here's a map of the ponds, and I did my first sort of serious scientific study of the smooth newts in these ponds. They were a series of concrete lined ponds with a little bit of a stream flow going through them, surrounded by bushes and other undergrowth, but slap bang in the middle of the college campus. And I got very interested in newt behaviour, and I thought it'd be interesting to look at the daily activity profile of the smooth newt. And to do this, I became something of an insomniac. So what I did, I used to get up early in the morning, just before dawn, rather about 5 a.m, go out to one of these ponds, and I'd put a strip of bandage down through the bank and along the bottom. It was quite a shallow pond. And I'd sit by the pond and I would count the number of newts that I saw crossing this piece of bandage for 10 minutes. And then I'd come back 2 hours later and I'd repeat it. I did this right through the day and the night, up until about, I think it was 3 a.m. in the morning. So I pretty much stayed awake for 24 hours. And I managed to construct this activity profile for the smooth newt. So you can see from the graph here that the number of transect crossings increased dramatically at dusk, and then it sort of fell off during the night. And then I also counted the incidence of sexual behaviour, again, stimulated by the work that Tim Halliday had done. He described the courtship dance of the smooth newt in immense detail. So I counted the number of sexual encounters that I saw, and you can see that that peak corresponds very clearly with this dust peak. And then I counted the number of times I saw them swimming into these little clouds of zooplankton and taking snaps at the zooplankton. And I think what was intriguing was that they seemed to have these two things they do after dark. At dusk, they seem to be mainly engaged in sexual behaviour. Then they stop chasing each other and they start feeding. Now, as I'm sure everybody realises, that throughout the animal kingdom, having sex is quite an activity that causes you to lose your breath. And I counted the number of times that smooth newts came up to the surface to take a breath of air. And you can see that that also corresponds quite neatly to the time when they're showing most of their sexual behaviour. And as Tim, I think, I've very articulately described, he says male smooth newts really undertake their courtship while they're holding their breath, and they face a bit of a dilemma for how long they can hold their breath for. Probably is related to how long they can hold a female's attention for. The other thing I did at these ponds was that there are a series of stones around one of the bottom of the ponds here, and I used to go out and turn these stones over and count the number of newts that I actually saw. And here's a graph of the number of newts, males and females, throughout the year in the water. And I also counted them, the numbers of newts that I found on land. And as you can see, there's a lot of newts on land during this aquatic period as well. They're not all in the water at the same time. But the intriguing thing was this period pretty much between the end of November and beginning of March, when there were practically none on land. There was a little bit of a blip in January, but effectively they disappeared entirely from these terrestrial refugia during the winter months. Now, if I roll on to 40 years later and Kent, and also roll on from a bit of scientific data to pure anecdote, I live in the village of Wye in Kent, and most days of the week I'm taking my dog, Cairn Terrier Ted, for a walk around the village. And Wye has some nice ponds in some of the gardens, and these are two gardens here where there are ponds. You can see the one in the photograph on the left, and the other one with the brick wall. There's a large pond behind a hedge here. And I use as my barometer of newt activity as whether I can see newts active on the pavement. And I would say on practically any mild, damp night during the winter, I can usually see newts that are active. And what has actually struck me is that newts are possibly not shutting down and hibernating like they were 40 years ago. They're probably pretty much staying active right through the winter, as and when the weather allows. And I think these mild winters are therefore becoming a bit of a problem for newts. Jumping forward again, 1983 to 1986, I'd sort of finished what I might call my apprenticeship. I'd finished my degrees at the University of London, and I got my first proper job, which is a research fellowship at a field station run by the University of Wales in mid Wales. And it was a marvellous 3 years, because under this research fellowship, I could pretty much do what I liked. I mean, when my wife is out of earshot, I say these were the 3 happiest years of my life, because it was really being paid to do, what your passion was. And as you can see here, this photo here, the haircuts have evolved a little bit since the 1970s, but still leave a little bit to be desired. But a colleague that I'd been working with had devised these little pitfall traps for invertebrates, from cutting a squash bottle in half and putting the funnel in the top. And I looked at these and I thought, I wonder if you could catch newts in those. And I tried them out and they worked a dream. And my main interest at this time was working on the relationships between smooth and palmate newts. And what we understood about these two species is that generally the smooth newt is more of a hard water species, and the palmate newt is more of a soft water species, but they do overlap in their distributions in many places. And Llisdinham Pond, which was right next to our field station here, and very intensively studied through toads and the frogs as well, provided a real interesting opportunity to sort of try and work out if there are any differences between these two species in the pond. So the bottle traps that we used, we weren't using them for surveying, we were using them to look at the distribution of newts in the pond. And what I'm holding up in this photograph is a string of bottle traps that we used to monitor the distribution of newts at different depths. And to cut a long story short, there was absolutely no difference in the distribution of newts in this particular pond. They use the same microhabitats, the same areas of shoreline, the same reed beds. And my 3 years in Wales, I think the conclusion was that there wasn't really any niche partitioning between these two species where they coexist together. Round about this time, I also got more interested in great crested newts, because great crested newts in mid Wales were on the edge of their range according to the distribution maps. And we did a number of wider surveys in mid Wales. And the interesting thing was that crested newts started to turn up in some quite unlikely looking ponds. They often occurred in these upland moorland ponds that were devoid of trees, sometimes slightly on the acid side. And the interesting thing was that we only detected these here with the assistance of bottle traps. If you went round with a torch, you'd never actually see anything because they were very low densities. Many years later, we came to look at this a little bit more systematically, and we compared mid Wales with Kent. And we found that in Kent, really bottle trapping as a very general rule, and there are pond-specific exceptions, but bottle trapping doesn't really add to anything that you might not see by torch counting. If you find them by torch counting, then you're likely to get them by bottle traps. Whereas in these ponds in mid Wales, torch counting seemed to often fail, but we pick them up at low densities by the bottle trapping. Over the years, I've published quite a few pretty obscure herpetological publications, but probably one of my most obscure was published in this journal, The Transactions of the Radnorshire Society. And this was very much a provisional set of distribution maps for Radnorshire. The Radnorshire Society is still going, so I looked it up on the internet. It focuses on archaeology and culture and natural history. And they asked if I could write an article for their journal on the distribution of reptiles and amphibians in Radnorshire. And I said, well, we don't really have much information. But anyway, I compiled these not terribly informative tetrad maps for all the species we have in Radnorshire. And I really think this is probably one of my least read articles. They asked for some photos, and I provided some photos of some herps, and the photograph of the slow worm actually appeared upside down in the final journal, which I was a bit worried about originally, but then I thought, well, probably nobody's ever going to bother to read this article anyway, so it probably doesn't matter much. Rolling the clock forward a few years, 1989, we saw the organisation of the first World Congress of Herpetology in Canterbury that was organised by the newly founded DICE at the University of Kent. And I should say, I wasn't actually at DICE at this time. I was, after working in Wales, I thought I was becoming sort of, the only strings I had to my bow was that I knew a lot about newts, and there are absolutely no jobs available for people who knew anything about newts. There was no consultancy or anything like that in those days. So I got a job as a lecturer at a college in Surrey, and I got roped into helping with the executive committee of the First World Congress of Herpetology. You can probably just see me down here at the front wearing a tie with a different hairstyle to the previous decade. But following my involvement with that, I got more involved with the University of Kent, and I managed to secure a 5-year research fellowship that took me to Kent in 1990. Now, at this particular time, the things that were really topical on the research agenda was global warming was becoming very, very topical. And the other thing that was still very topical was acid rain or surface water acidification. And I thought it would be interesting to put together a research project that would combine these two things, global warming and surface water acidification. And I put together this proposal with the rather pretentious title of Global Warming and Surface Water Acidification, Their Role in Structuring Amphibian Assemblages. And I managed to get some funding from NERC. If you translated the title of that project into more simplistic terms, what it boiled down to, I was looking at how does water quality and temperature affects the eggs and the larvae of the newts. And I was particularly interested at this stage of any differential effects between the smooth and the palmate newts. Was their distribution linked to differences in things like pH and water quality? And we devised a system for investigating this. This is Peter de Vija, who was one of our early students who came from the Netherlands, demonstrating what appears to be a very fetching plastic skirt. But in fact, this is one of our egg traps. We shred these bin bags and construct these and put these in ponds, collect newt eggs. And then we did a variety of experiments at a whole range of different scales. We did what I call the sandwich box experiments, where we raised newt eggs and newt larvae at different water conditions. We did experiments using what we call mesocosms, which are just really like mini outdoor ponds, where we can replicate things such as water depth, to a certain extent water quality, and so on. And then we did experiments in ponds by putting enclosures in ponds and raising newt larvae in different species combinations in enclosures. And this was an experiment we did in Kent, and this is one of the ponds down in Wales. I won't go into any of the details. The results of this came out, no real big surprises, but probably one of the more intriguing things was that, for example, this pond that I'm standing here in Wales, this was quite an acidified acid bog pond, a place called Corsif Lynn, that had a pH of about 5, and it had palmate newts in it, but no smooth newts at all. And we tried to answer the question, is that because they can't survive under these acid conditions? So we put some smooth newt larvae in these enclosures, and they grew just as well as the palmate newt larvae from the same site. So one of the conclusions was that they're not excluded from these ponds by water quality, they're excluded by something else that might be going on. Perhaps they haven't found the pond yet, or there's something else that's more subtle going on with the water quality. Now, jumping sideways a little bit, the foundation of KRAG. Now, I don't know if KRAG has anything in the archives about this, but I can distinctly remember going to the inaugural meeting of KRAG, although I can't remember exactly when it happened. I think it must have been about 1994, 95 or something like that. And it was really down to this gentleman here, who was a very lovely man called Paul Buckley, and he was the conservation officer for Kent Wildlife Trust. And Paul was phoned round a few people who he thought were interested in reptiles and amphibians, and said, would you like to come along to a meeting to discuss the formation of a group? So I said yes, and in fact we had the meeting in a room on the University of Kent campus. And thinking back, I think there were about 15 to 20 people at that inaugural meeting, and there are a few people there that I think are worth highlighting because they've gone on to do some very interesting things. One of them was Anne Riddell, who became the very first education officer at Wildwood. And Anne Riddell did some amazing work on slow worms at Canterbury Environmental Education Centre, now closed. And she was one of the first people to look at home ranging of individuals. And unfortunately, she never published this. It's only available in her thesis. But it's a wonderful piece of work because Anne was a journalist by training, so it's beautifully written. And the other thing that Anne did was edit, I think was one of the earliest KRAG publications, which was on the slow worm, and she got a range of people to write a number of articles. Anne was absolutely superlative at getting people to write things for her, because she was so charming, and you'd think, I'll have to do this for Anne because she's so wonderful. And Anne is now retired, but she's still doing some training courses up at Wildwood. Another person that was at this meeting was a long-standing friend of mine, somebody called Lee Gillett, who I knew before I came to Kent because she was involved with the British Herpetological Society. And Lee Gillett, he was a teacher at the Archbishop's School in Canterbury, a teacher of maths, and he was an amazing newt keeper. He used to travel all over Europe in his holidays to study and collect newts, and he had an amazing encyclopedic knowledge of newts and their distribution. And back in his home at Tyler Hill, he had a number of ponds in his garden where he had all the native species plus alpine newts, and he constructed 3 outdoor enclosures. One that was his Iberian enclosure, one was his Italian enclosure, and one was his Balkan enclosure, for the different species of newts that you get in those parts of the world. So, and all these were animals that he went out and collected himself. not long after I got to know him, his garden was getting rather cramped with newt enclosures, and he wanted to expand it. And the house next door came on the market, and he purchased that, not so much because he wanted the house, but he needed the garden space to put his ponds in. And he'd also got some wall lizards. So he built a wall for some wall lizards that he introduced there. And the downstairs living room of his new house next door, he converted to a desert habitat for desert reptiles and amphibians. So anyone walking up through Sleepy Tyler Hill and peered into the living room of this downstairs house wouldn't see a three-piece suite and a TV set. They would see a complete landscape desert environments with banks of UV light and some desert toads and lizards hopping around. Lee was very useful to me because in 1996, I wrote this book, Newts and Salamanders of Europe, for which I required some photographs of European species. And because Lee had every single species of the 12 species that were then known in Europe, I photographed his newt collection, which was pretty unusual. I don't know, there's no other way I would have managed to get the photos of all those species. The other person that was at the inaugural KRAG meeting was Derek Fraser. Now, Derek Fraser, I mean, he'd retired by now, but he was a naturalist who lived near Maidstone, and he used to work for the Nature Conservancy Council, the precursor to today's Natural England. Very distinguished herpetologist, done a lot of work on newt distribution, and also published the second New Naturalist volume on reptiles and amphibians. Derek was an interesting individual. He had an amazing ability to nod off during meetings, even if they were meetings that he was actually chairing. But he would sort of manage to sort of come around and contribute an articulate and poignant point to the conversation, even when he was sort of been asleep for the past 5 minutes. And then after that, he'd nod off again and then come back round, sort of resurface into the meeting a few minutes later. So very, very interesting individual. So what happened at that meeting back in the 1990s? Everyone chatted a little bit about what they were doing. And then Paul Buckley valiantly said, right, we need to form a committee. Who's going to volunteer? And at that point, a lot of the people, including myself who was present, looked at the floor and shuffled our feet because we all thought, well, I'm too busy to get involved with the committee. But Paul valiantly persevered, and two people who put their heads above the parapet were Lee Brady, who was then a young student, and Renata Plattenberg, who was also a student at the time. And I think they were very instrumental in getting this embryonic KRAG off the ground in those early days. And yeah, those early days, I remember there were ups and downs and there were personality clashes and various other things. But I think it's sort of very interesting to see how KRAG has developed. And, you know, as I think Steve alluded to, it's a very successful ARG, and I would go even further than what Steve said earlier. I would say KRAG is undoubtedly the best organised and the most active amphibian and reptile group in the country at the moment. So that's a little bit of a diversion. So when I was in Kent, another place I got involved with was Wellcourt Farm. And this was really down to Lee Gillett, who knew the farmer. And we started what was going to be a 3-year PhD project looking at the newts in these 4 ponds at Wellcourt Farm. And the PhD student was Claire Williams, who is still around and now works for Kent Wildlife Trust. And effectively, we surveyed these ponds on a weekly basis. We captured the newts, we photographed their bellies, and we looked at the individuals and the population changes over time. So this is a map of the 4 ponds. Down at the bottom here, we had this pond that we called the swimming pool. For some strange reason, in the 1960s or 70s, the farmer decided to build a swimming pool on the edge of a woodland, probably the worst possible place for a swimming pool. And it was also ridiculously deep. It was about 2 meters deep at both ends. So it was rapidly abandoned as a swimming pool. The most important ponds in the system were these ponds that we called the garden ponds, which were constructed in the 70s from butyle liners. And then there were 2 other ponds, which were more traditional field ponds called the Pyland Pond and the Snake Pond. over the years, we worked out that the swimming pool was absolutely no successful breeding. Adult crested newts were in there, but effectively it was a predator soup. It was full of beetles and dragonflies, and a lot of the eggs got infected with fungal infection, and it was complete sink population. The garden ponds were managed quite well, and there was successful breeding there every year. And the other two field ponds had intermittent breeding success over the years. What we did work out is that there was very little adult dispersal between these ponds, less than 1% of the adults moved between them. But juvenile dispersal was a little bit higher. So probably the main mechanism for colonisation of these ponds was through movement of the juveniles. If we look at the population size of the crested newts over the years, it's a case of up and downs. The heyday for the crested newts was around the turning of the new millennium, and then we worked out through statistical modelling that this was down to the level of successful breeding, the level of recruitment, and also mild winters were not good for crested newts. If you had a mild, wet winter, they didn't seem to hibernate as well, and they had lower survival. And then the second period, there were other things that came into play. But if you notice from this graph, which shows the profiles in the different ponds, that as we predicted, that the piling pond and the swimming pool both went extinct. So they were very much sink ponds. And the main driver of this whole meta-population was the garden ponds, which ironically was the smallest pond in the system, and yet it was the most productive. Jumping back to the university, we were continuing to use mesocosms, all these mini ponds, to do experimental work. And by this time, we had a few new people on board. Sue Young, who became head of conservation at Kent Wildlife Trust, Brett Lewis, who some of you know, and Amy Wright were all involved with this work in the late 1980s. And at this time, we were looking at sort of predation trials, and we weren't allowed to do any predation trials where things like dragonflies or fish actually fed on tadpoles. But what was more intriguing was how tadpoles responded to the chemical cues from predators. So this was an experiment where I think we had a dragonfly nymph in a cage, and we were monitoring the impacts of the behavior of the tadpoles on those. What we discovered was something that, as always, you know, it happens and you don't expect it, is that these experiments were quite complicated to set up. You spend a lot of time in this shed here counting out the tadpoles, and you set them up and they run for a couple of weeks. But we found that wild newts were climbing into these tanks and eating our experiments, which caused an awful lot of bad language when you spent days and days trying to set them up. But when we finished that phase of the work, we thought, well, this is actually quite interesting. We've obviously got newts in this area trying to colonize these ponds we've put in the ground. And so we said, let's look at this in more detail. So we sunk 4 very small 1 metre by 2 metre by ½ metre deep ponds in the ground in 1998. And we thought, well, just monitor these using bottle traps every week. And we've continued to do that ever since. And in fact, 10 years later, 2009, we put another 4 ponds in, and we've monitored all 3 species, great crested newts, smooth newts, palmate newts, who will colonize these ponds. And the remarkable thing here is that how the great crested newt population has increased over time. And we think this is in relation to the periodic draining of the ponds that we do. And what we've noticed is here in 2006, we drained the ponds for the first time. We then had 4 new ponds created, and then we drained them again in 2015. And if you drain the ponds, you effectively remove all the aquatic predators. And that means the following year, the newts have a good, successful period of reproduction. And it means 2 or 3 years later, you get an increase in the adult population. And I haven't updated this graph here, but I can say that the population has continued to increase. And not only have we got more than 100 newts in those ponds, but last April, we caught more than, I think it was 102 newts in a single evening in our bottle trap. So the population size is probably now nearer 150. It also allows us to keep track of the comings and goings of individuals. I sometimes say that what we do with these ponds is sort of study a type of newt. It's like newt EastEnders. you know, you get sort of Bruce the newt comes in for 2 or 3 years and then he disappears, and then miraculously he's back in the ponds again. And then we've got sort of females often seem to miss a year every now and then. And yet we've got people, you know, the Ian Deal of our newt ponds is Clint, who was around for many, many years. We used to catch him very, very regularly, although he sadly passed on. We didn't catch him after about 2013, but he lived for at least 14 years, which is much longer than the conventional age for newts. This has also allowed us to look at a range of other things, and one of the things that we've been looking at is crest development in the crested newts that we get. And the conventional wisdom is that, you know, crested newts come in the early spring and they haven't got a crest. They might have a small crest like this in early spring, and then they tend to grow it, and by the peak of the breeding season, sometime around about April or May, you'll have a nice male with a full crest. The interesting thing about these photographs is the dates they were taken on. This individual was photographed on the 19th of October, and the same individual had a full crest on the 21st of December. So I think there's some newts that are getting very, very confused about the climate. And we already have at least 14 newts in our crested newt pond, and a lot of them have crests that are actually bigger than this. And ongoing work is looking at how this crest development works in relation to arrival and departure times. What about the reptiles? I have to say, as you say, when I was a youngster, I was mainly interested in snakes, and it was probably down to my mother's flat refusal for me to have a snake that meant that I sort of got involved with newts and amphibians instead. But later in life, I sort of came to think a little bit more about reptiles, but it's fair to say that I've only been looking at reptiles for possibly about the past 20 years or so. And partly this was driven by a letter I received from this gentleman in the middle, Pat Gregory. And I got a letter. I knew of Pat Gregory by reputation. I was very flattered to get a letter from him because he's known as the doyen of North American snake ecology. He's based at the University of Victoria in British Columbia, done many years of works on rattlesnakes and garter snakes. And he wrote to me saying he was coming on sabbatical and he'd like to come to England for a few weeks in the summer. And he thought grass snakes might make an interesting comparison to some of the life history elements that he looked in North American grass snakes. And I read his letter and I thought it'd be fantastic if he comes here. But I did write back to him and try to put him off because I thought it just wouldn't work. And I said, the trouble is with grass snakes, if you're only here for a few weeks, they're really hard to find. They're difficult to catch. You'll get very little data in a short period of time, you know. And Pat wrote back to me and said, yeah, that sounds like the sort of animals I'm used to. I think this project might fly. And to cut a long story short, Pat came over every year for about 15 years until he retired a couple of years ago and studied the grass snakes at Fordwich and got some fascinating information. He did some radio tracking. He worked out there's a population, I think it's nearly 400 grass snakes at Fordwich. And the thing that Pat used to tell me some tales about people he used to meet, and there would be some, as you know, Fordwich, there's a lot of public access, a lot of dog walkers, and somebody walking a dog would bump into Pat where he's measuring a snake and say, good heavens, what have you got there? And Pat would say, well, this is a grass snake. And they say, well, I've lived here for 30 years and I've never knew there were any grass snakes here. And Pat, he didn't like using artificial cover objects, partly, I think, because at Fordwich, they would be subject to sort of public disturbance. But I think he was also a little bit of a purist and really thought that, you know, proper reptile people should use fieldcraft, which he absolutely excelled at. And I think what that did stimulated me to consider doing more reptile work. And also I thought this element of needing quite good fieldcraft really meant that reptiles weren't great for student projects, because if you haven't had a student who knew much about reptiles, they'd have to spend several weeks learning the method. But that, I think, really stimulated me to reconsider this. And we got involved with initially 2 sites in Kingswood. And again, this was through KRAG. KRAG had been doing a bit of work at these 2 sites. We call them the main site or the earthwork site. And then we picked up the baton round about 2005 And then a little bit later on, Steve Peters at Kingswood said, come and have a look at this other site that we're interested in. We've recently managed it, and probably Soakham Down, which we've been looking at since 2014, probably the best reptile site that we know of. So what we do at these sites is that we have directed transect routes and cover objects. This is Soakham Down, where we've got a combination of single and double pins. And again, to cut all the details out, we do about 10 surveys a year at different times through the spring and summer. And this has allowed us to do some sort of population assessment. So we've plotted here the population trends at Kingswood for slow worms at Earthworks and Soakham Down over the same time frames. Before 2015, I don't think there was much evidence that there was any change in slow worm populations at the Earthworks. But what is rather worrying is that these recent years, they seem to be declining, although I hasten to add that there's a whole range of things that might mask this. Perhaps they haven't declined. Perhaps it's just down to change in habitat and they're still there and we're not detecting them as well. So it's always a tricky one when you're just dealing with simple counts of animals. The adders allowed us to get a little bit more detail in the data. So just like Rick Hodges and his amazing site of Fackenden, we've been photographing heads of adders to allow individual identification. And this allows you to get much better population estimates. And this is the population trajectory for the Earthworks, and this is the population trajectory for the main site. And I think this is work that was done by Laurie Hill's MSc students. And I think the interesting thing here is that we also managed to get estimates of annual survival and detectability. And the annual survival, our statistical model said, wasn't varying over the years, so there was no decline in adult survival. And it also told us that the detectability, our ability to detect the snakes wasn't declining. On average at the Earthworks, the detectability was about 0.51, which basically means over a year, we were catching about half of the adders that were there. At the main site, it was slightly lower, about 30% of the adders. So in some ways, these more detailed analyses are control for these issues of survival and detectability and suggest there's other things going on with the adders. Soakham Down, I don't think we've been looking at for long enough. There is a slight downward trend, but I think we need more years. I suspect Soakham, I haven't added in the most recent data, but this trajectory has continued to go down. So I suspect there may be some issues with these adder population declines there. Again, survival 0.69, detectability, we're getting about 63% of snakes there each year. So, you know, why are things declining? Well, things decline when deaths outnumber births, or whether the animals moving out are higher than the animals moving in. So could it be reduced to reduce detection over time? No, we found that. Is it down to reduced survival of adults over time? No, not down to that. Could it be down to reduced fecundity? It could be. It could be that mild winters are causing animals to use up more of their energy, which is reducing their ability to reproduce. That in turn could produce reduced recruitment, reduced breeding success, and equally there could be animals moving away from the site as they become more unsuitable. And one of the problems at Kingswood is rapid succession to woodland. Because these are open sites in areas of woodland, this photograph here, back in 2012, if I had the same photograph, you'd see a lovely view over the Stour Valley. As you can see, it's now rapidly encroaching into birch scrub, and we are working with the forestry team here to try and keep on top of that. And again, here's a group of KRAG volunteers and some DICE students just pre-lockdown, I think, when we did our last log stacking exercise up at the Down to try and keep these areas open. So just to try and sort of finish off, I've talked a little bit about the past, I've talked about the present, and I've talked about the future. What might the future hold? Well, here's another very familiar face, not holding a gin and tonic in her hand, as you might think, but holding a jar of pond water. And one of the things that we've got very interested in is this idea of environmental DNA. And the thing about pond water is that it represents a complete soup of the DNA of the organisms that are swimming around in it. all living organisms will be releasing their DNA into the environment as they carry out their various activities. And there's two basic approaches to this. You can take a sample of pond water and you can run it through a quantitative PCR machine and then probe it with a known, say, crested newt DNA and see what you pick up. Or the more modern method is what we call metabarcoding, where you use next generation sequencing and you extract the DNA and then you compare it to what we call bioinformatic libraries. And this will tell you not just what species is in there, but the whole range of species. And in fact, although the qPCR method is the standard method for crested newt surveys licensed by Natural England, it is becoming a little bit antiquated as the scientific end of eDNA is moving much more to the metabarcoding. And we've done a little bit of metabarcoding on pond water, and it's quite interesting what you pick up in pond water. We've picked up Natura's bat DNA in pond water, so at some time there was at least one Natura's bat that flew over the pond and defecated into it and released DNA. You quite often get grey squirrel DNA and various bird DNA in pond water, again from overhanging trees. So this is a very powerful method, and I think in combination with some fancy statistics, if I'm honest, beyond my comprehension, you can learn a lot about the things that are going on within ponds, both on a local and regional scale. Our long-term vision for the ponds we're looking at the field site, and this was work started by Andy Buxton while he was a student postdoc, is that we can barcode all of the individual newts in our pond. And we thought, well, is it possible to identify all the newts by just taking a sample of pond water? And we've made some progress with that, but it's not quite complete. We're not quite there yet with the lab methods. You're looking at nuclear DNA, which is very, very, the concentrations are absolutely tiny and it's harder to amplify it and identify it. But what we do have is an interesting sort of family tree, or a provisional family tree of the newts in the pond, who give us some indication of who's been mating with who. And I can look at this picture of the newt in the middle and instantly know this is what we call the most famous newt in the world, Clint, because he's got this rather sad face on his belly with a zit on his chin. The other use for eDNA is in single and multi-species distribution models. So you can take species records, you can combine those with all sorts of interesting climatic and landscape layers, construct an interesting statistical model that will then give you a suitability map. And again, I think we're moving away from what we call single species models to what we call joint species models, that not only can look at the impacts of these environmental factors, but the interactions between the species that you have in these areas. And when you combine this with remote sensing data, such as satellite imagery, you can do things such as carbon flux within an environment, which is very interesting and useful for climate change. So this is, for example, a habitat suitability map for Kent based on eDNA and GLM statistical for great crested newts. Perhaps not surprisingly, looks pretty much like the KRAG distribution map that we have for great crested newts. It's a well-studied species, and this is just an indication of the sorts of things you can do. And possibly a next step down the line will be to try and look at how this relates to other species, if we can get pond samples and metabarcode them from a large number of sites. So that's pretty much what I had to cover in this sort of whirlwind tour of the past 4 decades. As I said at the beginning, this is not really a talk about what I've done. It's been a talk very much about the various people that I've had the pleasure of working with over the years. There's a very large number of them. This was one of the events where people got together. Some people from different phases of the past 4 decades met here for the first time, ironically. But I often say I've got the easy job because I usually sit behind the desk filling in forms and risk assessments, while it's really these people who are actually out doing all the work. But I hope there's some life in the old dog yet, and I hope I will be able to continue at least a few more years, although I suspect I might look more something like this by the time my days are done and I stop falling in ponds and stop being chased by park keepers and golfers. So that's pretty much it. Thank you so much for your attention and coming to the meeting, and always realise that there can be some very unexpected things that you can find in ponds if you spend 40 years doing herpetology. Thank you.
Richard, thank you so much for giving such an exciting and enthralling talk, not only about yourself, but and how you came into studying newts and progressed it further from there, but also giving us a view of the future as well. And we're extremely grateful for that. I'm sure there will be some questions. So I'm going to sort of hand it over and see if anybody would like to ask Richard some questions. So if you've got any questions you would like to ask Richard, sort of fire away. If you take off your mute and just ask the questions, off you go.
Richard, clearly KRAG is very concerned about trying to motivate people to show greater interest in our reptiles and amphibians. Remembering back to my own childhood, I couldn't really find very many friends who were interested in the same thing. And like you, I am very sad about the disappearance of the open air reptillery in front of the reptile house at London Zoo. I remember in the days when I was working at London Zoo as a holiday job, our comments about people effectively trying to fish the adders out of the reptillery there and make off with them. So clearly there was some enthusiasm for reptiles. They wouldn't otherwise be doing that, I suppose. But obviously you've had an enormous amount of experience in a university context of the amount of interest there has been in herpetology. Have you seen a big rise in the interest in people specifically for herpetology over the years?
Yeah, I think there definitely has been an increase in an interest in herpetology, and I think that I think it's become a bit more respectable. You know, when I started, there was no we had the Wildlife and Countryside Act, but we didn't actually have the Habitats and Species Directive. So there was no what I call crested newt industry that we have here. There were no consultants doing this. The NGOs, there was the Herpetological Conservation Trust, which had a few staff, and then there was the BHS, which was entirely voluntary. So there were literally no jobs in herpetology. So you were regarded as something of a bit of an eccentric if you had these strange habits, and you tended to sort of seek out other like-minded. There were other I had a really good friend at school who was also as interested in me, and we used to do a lot together. So I think what has actually happened is that with the rise in the species protection in this country, and the fact that, you know, there's consultancies that are doing it all over the place, that people have started taking herps a little bit more seriously, you know, because they can hold up developments, and there's a lot more information about them, and we know a lot more about them than there used to be. So I think I think it has become a little bit more respectable than it was in you know, in my younger days. But there was always, as I say, I think the emphasis always was people on keeping things, even if you had a scientific interest. It was more of an interest in sort of doing observations on captive things, and you know, doing field work was pretty much rudimentary natural history. And I think we've got many more scientific tools to make a lot more of the sort of natural history data that is being collected now as well. So I'm quite optimistic, really.
Well, let's hope there's still animals to study.
Yeah, I think the problem is, yeah, in the, when I was younger, there were more herps out there. As I say, I remember, I don't know if there's any crested newts left on Mill Hill Golf Course, and I remember finding my first slow worms in the garden next to, I lived in North London, Haringey, which is like 3 miles from central London, and there were slow worms there. I'd be very interested to know if there's any slow worms that close to central London anymore. So I think there's always this case in mobilising interest and concern at a rate that can outstrip the issues that are being addressed. It always seems that you can have more people interested, but the issues are moving at such a rate that you still haven't got enough body, you know, mobilisation within the people who are interested to actually get on top of things. But, you know, you have to run the race to try and get there, I think.
Wow, fascinating.
Richard, you came into it very young. You're interested in sort of snakes and that. Do you still see that enthusiasm in youngsters, or have they got locked into the internet now and less so in actual going out into the wild?
Yeah, I think that's a really good point. I think there was a great interview with Chris Packham and David Attenborough about 3 years ago in the Radio Times, and they both bemoaned the fact that a number of things have changed how young people engage with natural history. And one of those is the health and safety culture, and that you can't go and climb trees because they're too dangerous, you might fall out. Or, you know, you can't go wandering around a park in case, you know, a strange person abducts you or something like that. And also there's been the collecting mentality has rather, you know, frowned upon. And I remember Chris Packham, as always being controversial, said, you know, if I saw a kid out and climbing up a tree to sort of pinch any bird's eggs, I'd give them a helping hand because that's how I started, you know. So there is that downside. And I think, yeah, the problem is it's all internet based now and it's all social media based. So there has been that change in mobilization. So I think possibly there's far fewer come through the traditional, you know, cheesy, corny background that I did. But there still is sufficient information. You know, I think the internet has a positive side because there's far more information out there. It's much easier to learn things through that than there has been in the past, I think. So I think it's a double-sided coin. And I've noticed we do get students through the university who do come explicitly to study herpetology. And, you know, Bangor University runs a degree course in herpetology. And, you know, there is a market for it, I think. It's difficult to know what proportion of the population is interested in it. So I think the drivers of young people's engagement with it have certainly changed. And I think they've changed in some ways for the better, in some ways for the worst.
Thank you. Thank you. Go on, Gal. You've got to unmute.
Thank you for the talk, and not thank you for that photo. With the eDNA, is there a kind of standardized rate in which it denatures, in which you can't pick anything up in the pond? And if there is, do you know if it's an equal rate of decay across species or whether it's different?
Yeah, it does decay and it's very variable. It depends on substrate, it depends on water quality, it depends on ultraviolet radiation, depends on initial eDNA concentration. But the work that Andy did when he was around, we found we could pick it up probably about 3 weeks after we knew there were animals removed, but there were certain types of substrates that resulted in it binding with it. So it will persist, but I think the conditions under which it persists vary from pond to pond, I think.
Thank you. That's really interesting, at least with, if you're looking at that, you know, it has to be a recent event if it decays at that kind of rate. So brilliant, thank you.
Yeah, I mean, eDNA, it is used in archaeozoology. You know, there are people who are looking in montane habitats and retrieving, you know, DNA that's thousands of years old from substrates. But, you know, different types of conditions, different types of preservation, different species. So, but as far as crested newts are concerned, I would say that, yeah, if you find DNA, it's an indication of the presence or recent presence of crested newts as a very general rule.
Yes.
Richard, really superb talk. You mentioned water quality several times. Does your examination of water quality extend to looking at chemical pollution, things like agricultural runoff, pesticides, things like that?
We haven't looked at chemical pollution specifically, although there has been quite a bit of work done on that. Rob Oldham in Leicester quite a few years ago looked at some of the effects of pesticides and herbicides. And again, the impacts are often context specific. So you can often have what we call lethal effects. In other words, very, very high concentrations, it can kill things, but more likely you're going to get what we call sublethal effects. And that is where you get a change in behavior of the animal. In other words, it won't kill them, but it might mean that they swim around more. It might cause hyperactivity in tadpoles, and therefore it can expose them to predators more often. So even one of the things is, even, you know, I think we have to be very cautious about accepting like lethal levels of concentration, because they might not be important. There might be very subtle things going on with pollutants that are causing sort of knock-on effects down the food chain, because what we do know about amphibians is they can be very sensitive to, you know, low concentrations of certain types of chemicals within the water, and that may change their behavior and change the predation competition dynamics and change what's there in the longer term, really.
Absolutely. Thank you.
Richard, can I ask another question? You mentioned about mild winters and the impact it's having on great crested newts and when their crest is developing. There seems to be a theme running through sort of this. Well, I'm thinking particularly of dormice, hedgehogs, bats, other sort of, in those cases, mammals that are hibernating. Is there a lot of research going into how climate variation is impacting on hibernating creatures at all that you know of?
Yeah, I think there are bits. I mean, not so much on amphibians, but the conventional, you know, we're dealing here with species that have evolved in a cool, temperate climate, and they've evolved to shut down over winter. So the reason they shut down over winter is because there's insufficient food around. So if you play around with that and they maintain if they're semi-active, they will burn energy, but they won't be able to feed. So they will emerge in the spring with, you know, low fat reserves, which might mean that they're going to be unable to get through the following season. And I think it can also mean that for some, I think female crested newts in our population, a lot of them don't breed every year anyway. They breed every 2 years. And again, this is a common feature of ectotherms and cool climates, is that they can only get enough energy together to mature a set of eggs every 2 years. And if you have mild weather, it stimulates the activity and they burn that energy up, and they might even try to breed every year, but less successfully. So I think there are bits and pieces going on that. And I think with our crested newts, it's interesting, it's more complicated than what we've looked at the long-term data is the main breeding period, peak breeding period, is actually pretty stable. But the way climate change seems to be affecting it, it's the individuals at the beginning and the end of the season that you're getting the pioneer individuals that are coming in at odd times. The tails of the distribution are changing, and we're getting individuals that are hanging around longer. So I wouldn't say there's been a net shift to earlier breeding, but there are individual newts that are getting quite confused, and I think that seems to be also correspond with what we're seeing in other species as well. And I think what we might be, again, I don't know why the adders are declining, but a working hypothesis is that it could be a combination of mild winters, in that they're staying semi-active, unable to accrue enough energy to get them through the winter, combined with sort of habitat change. So I think a lot of these declines are probably due to double or multiple whammies rather than any single driving factor that work on them consistently, I think. But I say that's just really a working hypothesis rather than something I've got any data for.
So the climate variation changes. It's just another nail in the coffin, so to speak, yeah, rather than the great nail.
Yeah, I mean, amphibians as a group, I've always argued, amphibians were around before the dinosaurs went extinct. So as a group, they've been around a long time and they're quite resilient. But it's not the fact that it's the rate at which climate change is happening rather than the magnitude of it. So, natural selection operates over long generations. And if you've got things happening over 20 years, then it takes longer for species to adapt to that rapid level of change. But short term, we all know frogs often spawn in February and then there's snow in March, and then the frogs hunker down again and they come back again Yeah, they might have a bad year when they go underwater and they all die, but the population dynamics are adapted. Well, okay, but that will be compensated for a few years down the line when a few more come through the system. So you've got life histories sort of evolving to cope with these sporadic stochastic variations in climate. But when you have like persistent change, then it starts to sort of wheedle away at their whole life history, I think.
Super. Thank you, Richard. Do we have any other questions for Richard?
Sorry, can I just ask about water temperature? Because pond water remains a much higher temperature than the land around, and if it doesn't cool down early in the winter and is still relatively warm in the spring when the amphibians are arriving, does it encourage them to come out too early?
Well, I think it depends on the amphibians, because things like common frogs will usually hibernate. I mean, they can hibernate on land or in water, but you know, probably most frogs will hibernate at the bottom of a pond in water, and they will be at the bottom of a pond where the water temperature won't vary very much. Those amphibians that hibernate on land are probably much less sensitive to what's going on in the water, but they're more subject to, you know, how long it's been frost free and things like that. And it's always intrigued me that newts can be active at very low temperatures. I mean, as I say, last Thursday it was -2 in our ponds, and we had a thin layer of ice, and they were still active. We still got 5 crested newts in traps, even under a very thin layer of ice, and so they can be active almost down to freezing. And we know that common frogs have, you know, there's a lot of cool temperate frogs that have sort of antifreeze in the blood, which means that they can actually freeze completely solid. And then I think there's been some nice films of North American leopard frogs doing this. You find a frozen lump of rock that's a frog, and as it defrosts, it becomes active.
Yeah.
Antifreeze that stops these ice crystals forming in the blood. Whether that happens in newts, I'm not sure there's any evidence that happens in newts, but newts certainly can be active at quite low temperatures.
Thank you.
Okay, last chance for anybody who would like to ask Richard any questions.
One more thing I'd like to say is that I think we went through a phase, possibly the 70s and 80s, where children weren't encouraged to go out. There were too many dangers, but I've noticed the rise in forest schools, that the demand for forest schools is incredible. And I'm just really fingers crossed that this means young kids will have had that experience outside, messing about, and not be so fearful of the great outdoors, and perhaps it will be a little change in the interest in young people to spend more time being outdoors.
Yeah, I think you're right. I think there is I think that there's an acknowledgement now that, you know, well-being and biodiversity is crucially important, and there's an knowledge that wrapping kids up in cotton wool isn't going to do them any good. So I agree. I hope we see more of that. I mean, I used to go on holiday with my parents, and we used to stop to somewhere. I just used to wander off. They weren't bothered. They didn't know where I was going. And I just say, well, I'll be back in about an hour. And you know, I was probably 10 years old or 8 years old, and I just used to wander, you know.
Yeah.
I would also say that I've seen certainly at Vinters, I've seen a change during the pandemic. There's been when we were in lockdown, okay, people couldn't go anywhere else, but the numbers of people that were coming into nature,
I agree,
was quite high, and that seems to have kept going. There was a slowdown once in the summer of this year, but I think, Lynn, I agree with you. The forest schools are extremely popular, and the great thing is they come out suited and booted in any weather, and not just waiting till the spring. So I'm hoping we've turned a corner, and youngsters are finding a love of nature again, instead of a love of electronic gadgetry. So we'll have to see. Super. Right. Okay. I'm conscious of the time. Richard, thank you once again for a lovely talk, and it was great to see and great to hear the history of KRAG as well. And that was fantastic, and to see some of the photographs there as well. So thank you. So thank you very much. We do appreciate you coming in on a Sunday afternoon to give us your talk, and very appreciative. Thank you very much. So.